Okay, that's okay. When I get sliders, I usually get like either chicken or like if I go to Arby's roast beef.
Yeah, where is it?
Sliders, this is just for peaches.
Grab all your stuff.
I know.
All right, gang. So we did our Tinker Day today since Hollins has not gotten it together yet, but that's all right. We had fun. I know everybody should go hiking if you can. It's always a good thing to do. Hiking's always fun. I totally approve. Like I said, everybody is hypothesizing that they're going to do it tomorrow because the weather in Roanoke is really pretty tomorrow, and people have been why are you talking? People have been screen capturing like the Weather Channel and AccuWeather apps and posting it to all the little alumni groups. Like, look, I think it'll be tomorrow. So cool. All righty. So now we're going to talk about what we did in lab. Basically, this. Wow, you are hungry. Sorry.
Everybody's just watching.
That's like. That's sorry.
Anthony's like. It was very
Have you seen that really, really old YouTube video of the man that eats like a hippopotamus?
What?
It's really old. Have you seen it?
No.
Okay, that's kind of what that.
Now I have to go look it up.
Okay, if we have time, remind me, because you just reminded me of that.
It was falling. I had to get it.
I'm old.
I'm trying to be clean and not make a mess. Wow.
I'm just glad you're enjoying it. Enjoy life. All right.
This made my day like loud. Okay.
Mm-hmm. So we'll talk about RNA for a bit here. All right. So what's the deal with RNA? So what's the role of RNA? It's like DNA. It's going to be a nucleic acid that consists of a long chain of nucleotides, but unlike DNA, it's going to have three major differences. Can you think of any of them off the top of your head?
A is now U.
Okay, there's one. That's one. Can you think of any of the others?
No T's.
No T. Yeah, that's what he was saying. I think the same thing. No thymine. You got uracil instead. Think about structurally.
They're only single-stranded.
Single-stranded. That would be another one. So let's look. The other one is going to be obvious in the name. All right. So you've got deoxyribonucleic acid versus ribonucleic acid. So if you consider, if you think back to when we did chemistry, and those of you that were here, and we did all the different types of structural diagrams. These are your diagrams. Here's your ribose and your deoxyribose. So let me ask you this: they're sugars, right? Have you heard a certain term? What kind of sugar? When people talk about these bases here, they'll say it's a two-word something sugar.
Not nitrogenous base.
No. Five carbon sugar. You'll hear that. Okay, so what I want you guys to look at right here. See this shape here in the middle? What shape is that?
Pentagon.
It's pentagon. How many points does it have?
5.
Okay, and you get 5 sides and whatever. So when you see this, when you're dealing with organic chemistry in particular, that's 5 carbons. It's a 5-carbon ring. So there are all sorts of rings like this in organic chemistry where it's a certain number of carbons that are stuck together. And they have different shapes. And this is how you denote a 5-carbon ring, is this pentagon here. So you've got your pentagon and your pentagon. So you've got your ribose versus your deoxyribose. And you can see they're structurally different, right? All the little extensions are different. And then we also said RNA is generally single-stranded, not double-stranded. And then lastly, you guys brought up the most obvious one. You've got uracil instead of thymine. And you can see uracil and thymine look very similar, right? Very similar. So what are our functions of RNA? So the purpose of our RNA is to take all that information in our DNA library and make it something you can actually work with, right? The DNA itself isn't going to produce any protein. You have to have the RNA to make that happen. So you can think of it as a disposable copy of a segment of DNA, like a working copy or a single gene, kind of a single gene, kind of like when you write yourself a temporary note to go to the store. Like maybe you're gonna make cookies, okay? And you need to go to the store and buy certain ingredients. You don't usually take the whole recipe with you, right? You just scratch down on a piece of paper the things you need. That's how you can kind of think of RNA. It's this disposable copy. Its sole purpose is to be there for the whole transcription process and translation process. So RNA is only going to be involved when you're synthesizing protein. What's it mean to synthesize something?
Create.
Yeah, to create it. That's the whole purpose of your RNA, to synthesize protein. And then it's going to control the assembly of your amino acids into proteins. So remember, you've got amino acids. They come in little sets of 3. What do you call a chunk of 3? You remember?
The codon.
A codon, right. And then that codon is going to code for one amino acid. And the amino acids are going to stick together. They're going to be stuck together, built together inside a ribosome to create what we call this polypeptide chain. And then when you get the polypeptide chain, it's folded up a certain way. Ta-da, you have a protein. So your 3 main types of RNA. All right. First type you have is your messenger RNA. So what's your messenger RNA do? This is the part that's going to carry your information from your DNA to the other parts of your cell. So first off, where is your DNA housed? Where does it live?
In the nucleus.
Mm-hmm. Right. Where does protein synthesis take place?
Ribosome.
Mm-hmm. Are ribosomes in the nucleus?
Mhm.
No. Got to get out somehow. That's this dude. Okay. Next, you got ribosomal RNA. Okay, well, something has to make the ribosome. Ribosome's made of protein. Something has to make it. And something has to be there to, to create the structure. So this is where your ribosomal RNA comes in. And then lastly, you've got your transfer RNA. And this is what does your actual bringing of amino acid bases into the, nucleotide bases into the ribosome where they can be assembled from your RNA template. So how does your cell make RNA? So where's your RNA has to be made from what? Has to be made from, you have to start with your DNA. So it's going to be made inside your nucleus of your cell, right? So in transcription, segments of DNA serve as templates to produce complementary RNA molecules. The base sequences of transcribed RNA complement the base sequences of the template DNA. All right, you've got 2 big T words when it comes to this stuff. You've got transcription and translation. Have you ever had to do, well, today you did it in lab, but I would have said if you've ever had to write down a long number, you ever had to just try to, look back and forth and write a number down. Okay, this is sort of like how you're transcribing, you're getting your information and you're putting it down. You could also think of it almost like, well, it's just like what you did in lab today, but can you think of another analogy that's something that's similar to transcription aside from what we did in lab? So like in lab, we were taking a code and using sort of like a cipher, right? And we knew what the cipher was. It was A equals U, B equals C. It would be almost like if you were translating one language to another or symbols to another. Of course, Drew's like, the Zodiac Killer wrote in symbols. I'm like, there you go. Okay, yeah. So
Very true.
yeah, it's very Drew. So this is how transcription takes place. Transcription, you can actually think about it as actually writing, right? So you transcribe things if you're trying to copy information down. Like if you were writing that down in your notes right now and you messed up a word or skipped a word or something, that would be like an error in your transcription, but it would be you transcribing, taking that and putting it somewhere else. Translation is going to be different. So in transcription in prokaryotes, your RNA synthesis and protein synthesis are going to take place in the cytoplasm. Why would that be in prokaryotes? Why wouldn't it take place in it?
There's no nucleus.
There you go. There's no membrane-bound nucleus in the prokaryote. So if there's no membrane-bound nucleus, obviously your DNA is not going to be housed in a nucleus to begin with. So your DNA in prokaryotes is actually going to be like it's a ring. It stays as a ring, and it just sort of floats around in your cytoplasm because there's no nice, clean membrane-bound nucleus. So in eukaryotes, your RNA is going to be produced in the nucleus because that's where your DNA lives, and it has to be made from your DNA. So transcription is going to require an enzyme called DNA polymerase, and it's similar. Sorry, RNA polymerase, and it's similar to DNA polymerase. So we talked a little bit about DNA polymerase in a video a while back, I think. But your strands of RNA and DNA, they have what we call a 5' end and a 3' end. And all I want you to know is that if it's read, I think did we say this last week? If it's read one direction, For like the top section is read one direction and the bottom section is read the other direction. Right. So you start here with the 5 and you read to the 3. So you've got these things called promoters. All right. Here's where it gets real busy and sticky. So RNA polymerase is going to bind only to these things called promoters. And these are regions of DNA that have specific base sequences. So kind of like when we were doing start and stop, not all, like I was saying earlier, not all of your DNA information is useful. Not all of it means anything, right? So your RNA, when you're creating RNA, it's going to be looking for specific segments of DNA that are useful and tell it, okay, start here, or you need this information. And this is what we're calling a promoter. And it's going to say, all right, well, we're going to start here because this is where the useful information starts. So here's Amoeba Sisters, which everybody should love because they're amazing. And it's a really great.
It feels like DNA gets all the credit. Yes, the structure of DNA is amazing. It has a beautiful double helix structure, a twisted ladder. It has its own emoji. DNA stores genetic information and codes for your traits. However, sometimes what gets left out is how important RNA is. Without RNA, you actually couldn't get that genetic message out to your cells so that they can start producing proteins. We talk about this in protein synthesis. RNA is a very important biomolecule, just as important as DNA. In fact, RNA is even hypothesized as coming first, before DNA, in the RNA world hypothesis, something we need to make a separate video about. So let's compare and contrast DNA with RNA. You will find DNA and RNA in all living organisms. In eukaryotic cells, DNA tends to be found in the nucleus, while you can find RNA both inside and outside of the nucleus. Prokaryotic cells don't have a nucleus. Both DNA and RNA are nucleic acids, which are a type of biomolecule. Nucleic acids have a monomer, which, if you remember from our biomolecules video, a monomer is a building block. The monomer for nucleic acids is a nucleotide, so both DNA and RNA have nucleotides. The nucleotides of both RNA and DNA have 3 parts: a phosphate, sugar, and a base. Let's draw 6 nucleotides of DNA and 6 nucleotides of RNA. DNA is generally double-stranded, and if focusing on these 2 strands of nucleotides here, you can see they run anti-parallel to each other. RNA is generally single-stranded, so you're just seeing one strand here. The sugar in DNA is deoxyribose, and the sugar in RNA is ribose. This makes sense because DNA stands for deoxyribonucleic acid. That's helpful to know because the deoxyribose is a sugar, and nucleic acid is that type of biomolecule it is. RNA stands for ribonucleic acid, as its sugar is ribose. The bases in DNA are adenine, thymine, guanine, and cytosine. It helps remember the popular mnemonic apples in the tree. That helps you remember that A goes with T, and car in the garage. So C goes with G to understand how DNA bases pair. The bases in RNA are adenine, uracil, guanine, and cytosine. Notice the different one? It's uracil. So you have to change that popular mnemonic device here. Instead of apples in the tree, maybe the apples are under. Car in the garage still works though. We mentioned earlier that DNA codes for your traits, but it couldn't do that without RNA's help. In our protein synthesis video, we talk about 3 different types of RNA and their very important roles. mRNA, which stands for messenger RNA, mRNA's job is to carry a message based off of the DNA. In eukaryotic cells, DNA generally stays in the nucleus, but mRNA has the ability to leave the nucleus to take the message to a ribosome. Ribosomes make protein, and RNA is actually a major component of ribosomes. This type of RNA is called rRNA, which stands for ribosomal RNA. Finally, we discussed transfer RNA, more abbreviated tRNA. Its job is to transfer amino acids to match the correct mRNA codon. Codon charts using mRNA codons have been developed so that you can actually see which amino acid is brought for each mRNA codon. When those amino acids are joined together, they make a polypeptide chain. Proteins are made of one or more of these polypeptide chains, and proteins have tons of different roles. but we don't want to spoil it. Check out more in our protein synthesis video. Before we go, let's try a little 3-question quiz, shall we? Just pause it after the question so you have time to think about it. Question 1. If I have 8 DNA nucleotides, how many DNA bases do I have? And also,
How many base pairs? All right,
8 and 4.
8 and 4. Anybody disagree? Everybody's agreeing. All right, I like it.
how many base pairs? The answer, each nucleotide, regardless of whether it's a DNA or RNA nucleotide, has a phosphate, sugar, and a base. So 8 DNA nucleotides would have 8 bases. DNA bases pair like this, and that's 4 DNA base pairs. Question 2. If one strand of DNA has these bases shown here, ATT, GAC, can you complete what the complementary DNA bases would be for the other DNA strand?
So DNA to DNA. What do we have?
T, A, C, T.
You guys are good.
The
Yep.
answer. So remember those base pairing rules for DNA and the popular mnemonic. If placing the bases in this image, reading in the direction shown on this image, they'd be TAA, Question 3. In a process known as transcription, a complementary RNA strand called messenger RNA has to complement the DNA. More about that in our protein synthesis video. So if I still had the original portion of DNA here, what would be the complementary RNA bases?
Okay, RNA now. You. You guys are good.
The answer. So remember, it's asking for RNA. No thymine here, it's uracil instead. Apples under helps remember A with U. Car in the garage helps remember C with G. So in this portion, reading in the direction shown on this image, we have these RNA bases. Notice the answer is similar to the last answer, but there are uracil bases here instead of thymine. One last thing. Our models here, they're unable to show the beautiful 3D shape, the exact numbers of bases returned, the
She did her
but
best.
we've left you some links to explain. The goal of our videos is always to introduce the topic so that you can discover all the fascinating details and exceptions. Check out our further reading description to keep that curiosity going. Well,
It asked there at the end whether or not DNA is always a right-handed molecule. When you get into higher level chemistry, there are right-handed and left-handed molecules. It's really complicated, but it's kind of fun. Yeah.
What did they mean by DNA is generally double-stranded and RNA is generally?
So with DNA, you've got, it's like a ladder, right? And you can think of it like a ladder that's twisted, but it's actually even more complex than that. Like the rungs don't line up exactly that same way. It's hard to conceptualize because it's an interesting 3D shape. But you've got one side rung of the ladder here and one here, right? And these are your parallel, what would you call that part? These are the rungs. So these are what? I don't know. Those pieces, the long pieces, right? So your DNA's got 2, right? And then it's got your little base pair, your little bases come off of it like this, right? And there's another one here, and then they line up and go together. But your RNA is just one half of that. It's like you took it and cut it right down the middle. So it has your strand with all of your bases on it, but it doesn't have a pair. There's nothing to pair it with.
So what's
Is that what it's called?
So what's the double-stranded RNA?
So there isn't a double-stranded RNA. It's just DNA. DNA is double-stranded. It's got 2, those big long parts, and then your RNA's got 1 because it's basically just a template.
One stranded RNA pairs with half of a double-stranded DNA.
There you go. So ribosomes and protein synthesis. So the genetic code, what's the genetic code? How is it read? So your genetic code is read 3 letters, right? We say letters at a time. And what did you said it before, Michael, what did we call the 3 letters together?
Codon.
It's a codon, right? So each word is 3 bases long and it corresponds to a single amino acid. So we sat there at the lab and we had this cool looking chart, right? It looks exactly like the one I'm wearing, where you can literally follow it and tell you exactly what it's going to code for. Put this with this with that, and I get this. So your proteins are then made because you're going to take all of these individual amino acids that you're building with your different codons and you're stringing them together. When you string them together, it's your polypeptide chain. And your polypeptide chain, either by itself all folded up or combined with other polypeptide chains all folded up, is what's going to give you your protein. Now remember, what do proteins do? Like what's the purpose of building the protein? What's something that might result? Pretty much anything, right? You're talking about everything from your hair color to the structures that make up your nervous system and your neurons in general. These are all protein built. So it's really, really essential that you be able to build proteins. Now, here's what we're going to talk about in a minute. And this is an interesting thought, but all of your cells, all of your somatic cells, non-gametes, non-sex cells, they've all got the exact same DNA, right? Like if you dropped a skin cell on the floor and they found it and analyzed it, it would be the same as them finding your bones and being able to dig you up and go, oh, this is Cole because I can match his DNA from this bone cell to DNA left on his toothbrush, right? It's the same DNA in every cell. So how come your skin cell isn't growing lung tissue? And how come the cells that make up your cornea aren't growing hair out of your eyeball?
Because it only takes this certain part of the photo.
It has to do with, well, all the codons are there. Basically, it has to do with how your genes are regulated and expressed, which is really, really complex. But your DNA has a code within it to tell it how to use the code so that certain cells differentiate and do only certain things. So the basis form this language, your genetic code, which is your 4 letters. So what if I had GUT? What amino acid would I be producing? Can you tell? GUT. Yeah, valine. Okay. And how about if I had UAC?
QIR.
Yeah, that's gonna be tyrosine. So you can see, the interesting thing is, there was a question in the lab about if you had one code versus a different code, would it change your outcome? And it didn't, because they all coded for the same. Like in this case, where you have valine here, right? You could have GUT, GUC, GUA, GUG. It would all code for the same amino acid, right? But look here, you have to have U, G, and G to get this one. And you have to have UGA to get this one. It's the only ones. It's the only way it's going to work. So some of these are going to be way more specific than others, obviously. And it's just kind of interesting how you can come up with different combinations and sometimes produce the same thing. Others are much more specific. So each 3-letter word that we were talking about is a codon, right? So you probably saw this one in your lab. Methionine, that's AUG, and that's always going to be start. That is always your codon for start. And it's the same. I mean, that's just real. That's honestly what's in your DNA. Anytime AUG shows up, your mRNA goes up, time to start working. So following the start codon, your mRNA is read 3 bases at a time until it reaches one of 3 different stop codons, which end the translation. So here's an example of one here, which is UAG. There are actually 3 different ones that can code for stop. So this is all transcription. We were talking about transcription. The other one I said, the other T is translation. So what you need to remember is transcription is taking the message in the DNA and making your temporary copy. It's like writing notes. Translation is actually taking that information from your temporary copy and producing something with it. So it's very similar to like I said, you go to the store and or a lot of people use a library reference. You can think of your DNA in the library, and the library is your nucleus. And it's one of those reference books that you can't check out, right? You cannot check out the reference book. All you can do is go in there and make a copy or handwrite a copy of what's on the page, because it's not allowed to leave. You know those books that can't leave the library, right? So that's exactly how your DNA is. You take it out, now you're mRNA, right? You take it out into the world, and then you take the instructions that you found in the reference book and you use it to build the shelf you wanted to build. And that, you got the instructions for building the shelf in that reference book. So now you've taken that information, you're going to build something with it. That's translation. So translation is the actual building or assembling of the proteins. So this happens in your ribosome. So your DNA has left the library, has left your nucleus, Now it's gone through the cytoplasm. It's found itself a ribosome. So ribosomes use the sequence of codons in mRNA to assemble amino acids into what we call the polypeptide chain, right? And the decoding of mRNA message into the protein is the process we call translation. Okay, so what's the difference between transcription and translation?
Transcription takes the information, translation turns the information into stuff.
There you go. Transcription takes the information from the DNA to RNA. Translation takes the information from the RNA and builds proteins. That's exactly right. So you've got steps. Messenger RNA is transcribed in the nucleus, right? And then it enters the cytoplasm for translation. Each tRNA molecule has 3 unpaired bases called an anticodon, which is complementary to the mRNA codon. So they showed it like a little, it looked almost like a little cross or like a T, and it had 3 little bases hanging out on it, right? And it brought them in and it found the codon it matched and it kind of clicked them on. That's a really good graphic to show you kind of what happens here. So your tRNA is going to transfer these free floating, these amino acids are just free floating. How do you suppose you get them, by the way? How do you suppose you get amino acids? Well, yeah, that would be like how they would get into the nucleus or through it, probably more likely through active channels. But how would you get amino acids into your body to begin with? Yeah, you eat the protein, right? It breaks down into amino acids, which are the building blocks of the protein. Now they're free floating and your body can use them. It puts them where it needs them so that your transfer RNA can go, ooh, I need this one and I need this one, right? And it breaks them, it builds them from the nucleotide bases that it's broken the amino acids down into. Like they're all broken down into little letters like they started out, and it's rearranging them all. Like if you eat, think about it this way, if you eat a steak, right? You got to be able to take whatever proteins are in that steak, and there's going to be a lot of different proteins in that steak, but you got to be able to take those proteins and build completely different things with them. So it's like taking a Lego structure completely apart and building something completely different with it. Like think about babies. Okay. So like one of the main proteins in milk is casein. So if you're a mammal and you're getting predominantly casein protein, and you're getting other proteins too, but you're getting a lot of that, you got to build everything with that: hair, organs, teeth, bones. So you got to break that down into amino acids, break that down into nucleotides, free float them, rearrange them, make something different. Pretty cool, really, when you think about it. So we talk about this molecular basis for heredity. All right, so what's the central dogma of molecular biology? This is a good word, central dogma. What would that mean, do you suppose?
Main rule.
Yeah, kind of like your main rule. It's a rule to live by for molecular biologists. So your central dogma of molecular biology is that information is transferred from DNA to RNA to protein. That's how it works. DNA to RNA, RNA to protein.
All of molecular biology is described that.
Well, it's the foundation for it. It really is the foundational concept. Because if you don't, if you're dealing with a lot, molecular biology deals with a lot of protein, and you got to figure out how to get there. And this is how you get there. So gene expression is the way in which DNA, RNA, and proteins are involved in putting genetic information into action in living things. So expression has to do with, first off, what information is actually going to be activated. For example, is your cornea cell going to grow hair? But it also has to do with how much it's activated. Some genes are what we call upregulated, which means there's a problem and that gene is active too much. It's producing too much of whatever it produces, and that can cause issues. There's also downregulated genes, but expression is the way in which it's put into action. What's being expressed? How is it being expressed? So your DNA is going to carry information for specifying the traits of an organism. Obviously, that part's the kind of obvious part. Despite the enormous diversity in form and function, living organisms display remarkable unity at life's most basic level, which is the molecular biology of a gene. So your genetic similarity to a banana is remarkable. It really is. For real, because it's all the same. It's the same building blocks. There's only but so many. And this is your number of amino acids. That's it. That's it. So if this can build me, this can also build the ant. It can also build the banana. It can build Jack, it can build the pine tree outside. It's all the same stuff, just rearranged almost seemingly infinite number of ways, right? Which is really, really fascinating when you think about it. It's like it came to me once. It was kind of like, have you ever been to like a real diner? A real diner. And what I mean by a real diner is it has like a 20-page menu, but there's seriously like 50 ingredients in the whole place. They're just combining it every way possible. If you've ever been to a real diner, that's what it's like. It'll be like, we have a cheeseburger, but we also have a patty melt, but we also have chili mac, because it's the same ground beef and it's the same cheese and it's the same, they just combine things differently. And it's very similar. That's kind of how life works. So here's the Amoeba Sisters protein synthesis that they referenced. And this is a really good one too. All these are really, really good. They're great at summarizing and kind of nutshelling it all.
After learning about DNA, have you ever wondered how can the DNA actually result in a trait? Let's take an example, like eye color. Yes, your DNA has the genetic information that codes for the color of your eyes. Your eye color is based on a pigment that is inside the eyes. But in order to have that pigment, you have genes, which are portions of DNA that can code for proteins, which help make that pigment. So what we're going to talk about is how your DNA can lead to the making of a protein. This process is called protein synthesis. Synthesis essentially means to make something. So protein synthesis means to make protein. And you may wonder, what's the big deal about proteins? Well, you may not realize this, but proteins are kind of a big deal. They do all kinds of things. Proteins are involved in transport, in structure, in acting as enzymes that make all kinds of materials, in protecting the body, and so much more. You've got to make proteins. It's essential for you to live. And what is so cool is that you are making proteins right now as you sit and watch this video. It's happening in your cells. They're making proteins. So back to your DNA and its role in all of this. All of your cells have DNA, well, a few exceptions, and that DNA is in the nucleus. Some DNA is non-coding DNA. Some DNA makes up genes that are not activated. More about that in our gene regulation video, but we're going to talk about genes that are coding for active proteins. So how are we going to get the information from these genes out of the nucleus so that the cell can start producing the proteins that it needs to make? Well, let us introduce you to the amazing work of RNA. We have a video comparing and contrasting RNA and DNA. We'll be brief here in saying that RNA is a nucleic acid like DNA, but it has a few differences. Its role in protein synthesis also is huge. Before we get into the process, please note our typical disclaimer. We tend to simplify topics, but as always, we hope that you have the desire to explore this complex, amazing process later on, to learn all about the extra information that we don't have.
I love how they're looking at the horizon.
In protein synthesis, we can look at two major steps. One is transcription and the other is translation.
Right.
Transcription has a C in it and translation has an L in it. I remember that C comes before L in the alphabet, which helps me remember that transcription comes first. I like a lot of alphabet mnemonics. The transcription is when we're going to transcribe the DNA into a message. In your cells, the DNA is in the nucleus, so therefore we're doing transcription in the nucleus. In the step of transcription, an enzyme called RNA polymerase will connect complementary RNA bases to the DNA. These RNA bases are bonded together to form a single-stranded mRNA. The M in mRNA stands for messenger. Messenger RNA consists of a message made of RNA that has been based on the DNA. We do want to mention that this mRNA.
I want to show real quick, Jack, there's the answer to your, hopefully you can see it before they put that thing up. There's that. To answer your question earlier, so the orange is your DNA strand here, and it's been unzipped in the middle, right? From here to here. There's the unzipping, right? This little single strand is the RNA, and it's just coming in to build, and then it keeps moving on. Does that help?
Yeah.
There you go.
Or messenger. Messenger RNA consists of.
Yeah. There you go.
We do want to mention that this mRNA is not usually ready to go right away. There's usually a significant amount of mRNA editing that occurs. We highly encourage you to do some reading about that because it's
Little
not only
pencils.
fascinating, it's critical for the process to work correctly. So what's something great about being mRNA? Well, in eukaryotes, you get out of the nucleus. The mRNA can go out of the nucleus into the cytoplasm where it's going to attach to a ribosome. Ribosomes make protein. The ribosome is made of rRNA. And that's an easy one to remember because the R stands for ribosomal RNA. The ribosome is going to build our protein in the next step called translation. You know, you can find a lot of great clips and animations on translation that are just fantastic. We're just going to break down some basics of what's happening. In the cytoplasm, you look at this, you have all these tRNA molecules available. tRNA stands for transfer RNA. They carry an amino acid on them. An amino acid is the monomer for a protein. It's a building block for protein. Since we're making proteins, we're going to need those amino acids to build it. If you have a bunch of amino acids together, you can build a protein. So it's the tRNA that is going to bring those amino acids together to make that. But wait, how does the tRNA know which amino acids to bring? That's why the mRNA, the message, is so important because it's going to direct which tRNAs come in and therefore which amino acids are transferred. All of these tRNAs are looking for complementary bases. When they find the complementary bases on the mRNA, they transfer their amino acid. When tRNA is bringing in the amino acids, it reads the bases, represented by these letters here on the mRNA, in threes. So it doesn't read one letter at a time, it reads in triplets. That's called a codon. So for example, in this mRNA, the tRNA would read the codon AUG. One of these tRNAs contains a complementary anticodon, which in this case is UAC. All tRNAs that have the anticodon UAC will be carrying an amino acid called methionine. A tRNA with the UAC anticodon comes in to pair with the complementary AUG codon on the mRNA. It transfers the amino acid it carries, methionine. The tRNA will eventually leave, but it will leave behind its amino acid. That's the first amino acid before looking at the next codon. Before we do the next codon to carry this on, if you're wondering, how did you know that the tRNA that went with the AUG codon would be carrying an amino acid called methionine? Well, for that, you will find a codon chart helpful. You can learn to use a codon chart to determine which amino acid each mRNA codon will code for. Isn't it so fascinating that scientists have been able to determine which amino acid corresponds with these codons? I used to have a codon chart poster and just marvel at that. You can see on the codon chart that the AUG codon on the mRNA codes for methionine. AUG is also considered a start codon, as methionine is typically going to be your first amino acid in proteins. There are many types of amino acids in the codon chart, but there are even more possible codon combinations. That means there could be more than one codon that code for the same amino acid. For example, according to the mRNA codon chart, all of these mRNA codons here code for the same amino acid, leucine. That means their complementary tRNAs all carry the same amino acid, leucine. Okay, so going back to the mRNA, let's try the next codon on this mRNA, CCA. On the codon chart, you can see that codes for the amino acid proline. The complementary tRNA has the anticodon GGU. And look, there's the proline that we knew it would be carrying. The tRNA will transfer that amino acid and eventually leave where it can pick up another amino acid. These amino acids are held together by a peptide bond, and it will keep on growing. Typically, at the very end of the mRNA, there is a stop codon. Stop codons do not code for an amino acid, but when the ribosome reaches it, it indicates that the protein building is finished. So the result of translation is that you built a chain of amino acids that were brought in certain sequences based on the coding of the mRNA. But remember, that mRNA was complementary to the DNA. So the DNA ultimately was the director of the entire protein building. Of course, it couldn't have done it without some serious help from mRNA, rRNA, and tRNA. Protein folding and modification may occur, and the protein may need to be transported. This can all vary based on the protein structure and function. Another fascinating topic for another Amoeba Sisters video. Well, that's it for the Amoeba Sisters. And remind you to stay curious.
So it's good graphics. You can really kind of visualize it better. All right, so talk about mutations. Any of you guys have trouble with your coding today? You ever have something that made no sense? Yeah, occasionally something came up and you're like, oh. So that basically replicates a specific type of genetic mutation. There are different types. What you were seeing was what we call a point mutation. So mutations are basically heritable changes in genetic information. So these are mutations that involve, sometimes they can involve just a few nucleotides, and these are called point mutations. So that's what you guys, if you had trouble with the transcription today, that's what that was. That's a point mutation. So if a gene in one cell is altered, the alteration is passed on or can be passed on to every cell that develops from that original one. So this is a great device for understanding the different types of point mutations. And I'm grateful that some teacher somewhere substituted that, hey, come on, chicken breast, because that's not what was there in all of the original graphics. It's all over the internet, and it's a nice little cartoon human breast. So I found the chicken breast, somebody saved me some time. I didn't have to do that myself. So if you start with the word beast, okay, say this is your normal code. This is your normal word, your normal letters, right? Then in a substitution, you're going to have something like this, where you've got one that's been substituted for another. Now beast is turned to feast. You've got an insertion where you've added a letter, and this is where you get breast thrown in. And went from beast to breast because now you've thrown in something that wasn't there. Now this can happen if you're writing down a long number, everybody's done it. You transcribe it incorrectly. This is the sort of thing that happens here. You can have a deletion. Now we've lost a letter. Now we have best instead of beast. And then you can also have an inversion where they've been flipped. So now we have beats. Yeah, he's rocking away. So these are different types of specific point mutations. There are other kinds of mutations, which we're going to look at. These are all point mutations. So there are 4 types of chromosomal mutations: deletions, duplications, inversions, and translocations. So some of these are the same words, but they're different. So when you looked at the deletion and the point mutation, you lost a letter, right? That made up your word beast. Well, here you lose a whole piece of a chromosome. So this is a chromosomal deletion, which isn't the same as a point mutation deletion. A duplication, you can see right here, it's like what, green, yellow, red. And then in this one, it has it twice, green, yellow, red, green, yellow, red. And inversion, they're flip-flopped. It's like red, green, and then pink and blue stripes. Whereas this one, it's pink and blue stripes, and then green and then red. And then lastly, your translocation is when you've got one part of your chromosome that broke off and attached to another, and it like flipped around all funky. Now they're backwards and weird. And this is a translocation. So these are all things that can happen to your chromosomes. And obviously this is going to happen when you've got division going on, when you are replicating DNA and getting ready to go through your process of dividing a cell, and all of this stuff is supposed to be copied. It's, there are going to be chance mistakes because that's how life works. Not everything is going to be perfect every time. And some of these mutations, we all carry mutations. We all have thousands, probably more than that, in our DNA. And a great deal of them have zero impact. Some of them have a real negative impact. And some of them, it's not positive or negative clearly, but it's how you get variation in a species. And without mutations, you don't have any sort of evolution. You don't have any sort of change in species over time. That's what causes the change over time. So how do these mutations affect your genes, the way they are expressed? Well, like I was saying, some have little or no effect. Some are beneficial. Some can disrupt function. Yeah.
I'm gonna be an Alex here.
Oh, what'd you find?
Top how-to mutations.
Oh, wow. You found, look at that. Yeah, Alex would be proud of you.
Alex is watching this back like, oh, so proud.
Amazing, right? He's like, that's my girl. So the cellular machinery that replicates DNA inserts incorrect base roughly once in every 10 million bases. So small changes in genes can gradually accumulate over time. And some of them, it's just like one little change, and they call those single nucleotide polymorphisms, SNPs. Some of them are bigger, more obvious. And like, remember last week, we talked about how there are some genes that have more variability, because that's like a mutation at one point that got passed through certain populations more than others. And there's certain areas of the genome that are going to have more variability. And those are the ones they look at when they're trying to identify people, right? Because the rest of it's almost all identical, like I said, to a banana, you know? So mutagens. All right, so a mutagen is something that can cause a mutation. Sometimes it's chance, like we just said, but there are things that can increase the chance. So what's the most common way people get skin cancer? From the sun. So that's electromagnetic radiation, right? I mean, you get ultraviolet, not electromagnetic, ultraviolet radiation, electromagnetic radiation, X-rays and stuff. And well, yeah, same thing. So if you're getting ultraviolet radiation from the sun, it can cause damage to your DNA. Perhaps you damage the part of your DNA that tells your cell to stop dividing and die. And if that's a problem, that's how you get things like tumors, right? So look at some of these things. Who did we talk about last week? Who had cancer?
Was it Rosalind Franklin?
Yep, Rosalind Franklin got ovarian cancer and died. She spent her entire life working with X-rays, right? So that can do it. Now, when you go to the, I know, it's crazy. When you go to the dentist, when they go to take your X-ray, what do they lay on you first usually? The big old lead thing, like, right? You know?
And then they leave the room and tell you you'll be fine.
Yeah, exactly. They're like, we'll be back later. We'll be back. If you've ever had an X-ray at the hospital, they go behind that big lead wall and leave you in the room, but they leave you like, you know, you're all twisted in some weird position. They put like some foam block under your hip and they're like, don't move. And you're
Tellers is fun. They're just like, you'll be fine. We're gonna be behind this really thick glass,
Right.
behind this really thick wall.
so obviously a small amount of X-ray radiation isn't bad, but constant exposure is really bad. So if you work in X-rays, they're going to go and hide behind a wall constantly because they can't be exposed to it all day. And the dentist is covering up your body, but like largely they're covering up the most important organs that they're concerned with. They're concerned with, you know, all your reproductive organs in particular. Yeah. And yeah, all that sort of stuff. Like you don't want to be shooting X-rays into people all day long. And then there's other things like preservatives in foods. There's cigarette smoke, obviously bad stuff. Here's HPV. If anybody that's been past the age of 12 has had the discussion, or maybe not had the discussion, but we have the vaccine that they do now, the Gardasil vaccine. But HPV is human papillomavirus. It's a type of wart and it causes cervical cancer. So that's the type of mutation it tends to press. So that's one of the things that comes up with people is whether or not to get that vaccine. Obviously they're all optional, but it's one of the more optional ones, right? So yes.
I'm sorry, my brain immediately went to helicopter bacteria.
Helicopter bacteria. Right? Yes. That's funny. So without mutations, organisms cannot evolve. And because they are a source of genetic variability in all species. So some cancers are a product of mutations that cause cells to grow out of control. And the condition in which an organism has extra sets of chromosomes, you can get a whole extra set of chromosomes called polyploidy. Can anybody think of a condition where there's at least an extra chromosome? Yeah, Down syndrome. That's right. When we're going to talk about specific types of genetic mutations the next week or the week after. And that's one of the things that is discussed is a trisomy, which is instead of having one pair, 1, 2, you have a third of that chromosome. And in particular, it's trisomy 21 that causes Downs. It causes an extra chromosome that causes Down syndrome. There's also, I think it's 13, trisomy 13. I think that's the one. And it causes like brain development is incomplete. So you only get like brain stem and some cerebellum, but most of cerebrum is missing. Most of those children born with that don't survive very long. There are one or two that I know of that like are kind of in the public eye. I want to say it's like Rick Santorum's daughter or something is like way older. Most of them don't live through infancy, so it's kind of weird.
Is that all barbecuing or?
No, I mean, obviously,
Is it all
So-
benzoyl peroxide?
yeah, is it all benzoyl peroxide? So like, obviously, most things in moderation, you're going to be fine, right? But you want to be careful what you expose yourself to constantly and continuously. Now, if you live in California or if you visit California, they have little signs up in all their restaurants that talk about how if you have browned food, like golden brown food, like fries or something like that, that's a carcinogen because they have to label everything in California because it's California. Like it causes cancer to eat French fries because they've been browned and that produces, chemicals in foods because they've been carbonized to some degree. So here's mutations. We're going to watch a mutations video
Wow. I wonder if
from our
this is
favorite
something.
amoeba sister. I can watch amoeba sisters all day. Oh yes, Shannon.
I was gonna say the donuts wise. They're
Yeah.
cooked in like a golden brown. so
Yeah.
that counts still?
Of course.
Oh.
Does that give you a reason not to eat them?
Yes.
No.
Ever since I was a kid, I have been curious about mutations. I think certain movies may have had an impact. I used to imagine mutations with amazing
Or whatever.
abilities, but there was also a lot I didn't understand about mutations. Let me explain. Many people understand that a mutation is a change of genetic material. More specifically, a change within a nucleic acid. RNA and DNA are both types of nucleic acids. Therefore, anything with RNA or DNA can have a mutation. That means animals, and that definitely includes humans, also plants, fungi, protists, bacteria, archaea, they can all have mutations. Additionally, so can viruses. Many mutations can be neutral in effect. You can see this codon codes for the amino acid leucine. But if it experiences this particular silent mutation, even though a base has been altered, it still codes for leucine. It did not change the amino acid. Mutations can also be harmful or helpful, but it's important to understand that mutations are random. The organism can't will itself to get a certain mutation. We'll learn more about this in our natural selection video, where we
Love
talk about how a bacterium may already possess a helpful mutation that allows it to survive an antibiotic. But the bacterium didn't will itself to mutate to develop this certain mutation. Okay, so again, mutations are random, but there can be factors that make mutations more likely to occur. External factors like certain types of chemicals or excessive radiation, or internal factors like an event that causes a problem during DNA replication in interphase. During interphase, cells can replicate DNA before they divide. So let's discuss some different types of mutations. We'll start with gene mutations. DNA makes up genes, and genes can code for proteins that influence different traits. So when a mutation in DNA happens, which specifically means a change in one or more DNA bases, then different proteins can be produced, which can affect an organism's traits. In this example, we have fruit fly DNA. Mutations could include substitution, which means the wrong base is matched.
Oh, no.
Insertion, which means an extra base or bases are added in. There's also deletion, which means a base is removed. Insertions and deletions have the potential to be especially dangerous. Why? Remember in protein synthesis how we talked about how bases are read in threes? Well, if you add a base or remove a base, suddenly the number of bases total has changed. And if you read the bases in threes, depending on where it happened, everything that is read afterwards could be affected. We call this a frame shift mutation. If you look at this frame shift example here, where a single base has been inserted, you can see how the following codons, which have 3 bases each, are now all affected as the reading frame has been shifted. This example shows how it can lead to many amino acid changes. There are also chromosomal mutations. Remember that chromosomes are made up of DNA and protein, highly organized, and they have lots of genes on them. The human chromosome number is 46, with 23 from an egg cell and 23 from a sperm cell. This type of fruit fly here, however, has 8 chromosomes, so 4 came from an egg cell and 4 came from a sperm cell. Some examples of chromosomal mutations include duplication, where extra copies of genes are generated. Deletion, where some of the genetic material breaks off. Inversion, when a broken chromosome segment gets inversed. which means reversed, and put back on the chromosome, or translocation, when a fragment from one chromosome breaks off and attaches to another chromosome. There's more mutations than what we covered, of course, but the idea is that there are many different kinds of changes that can happen. If a mutation is going to happen, we already mentioned that there are especially vulnerable times, such as during DNA replication, but also there are other times too, like during meiosis. In fruit flies and other animals, meiosis makes sperm and egg cells that can have the number of chromosomes as the organism. However, sometimes those chromosomes don't separate completely. We call this non-disjunction. This can result in an egg or sperm cell that has too many or too few chromosomes. It's possible for a mutation to be passed down to offspring. Consider a protist with a mutation. Many protists reproduce asexually, and when they divide, the daughter cell can inherit the same mutation. A fruit fly, which reproduces sexually, can pass a mutation to its offspring if that mutation is found in the genetic material of the sperm or egg cell. You may wonder why we've been mentioning fruit flies so much. Well, they're awesome, but also it turns out that fruit fly mutations and how they are inherited are frequently studied. Leave some links in the video description for more info. These studied mutations can also occur in humans, and we'll get a gene mutation example of a substitution in the case of sickle cell anemia. First, a little background. Hemoglobin is a protein in your red blood cells that helps you carry oxygen. But in the disorder sickle cell anemia, the gene that codes for hemoglobin is mutated. If you inherit 2 copies of this gene, one from each parent, you can have this disorder. This disorder can make it difficult for your red blood cells to carry oxygen because the shape of the red blood cell is affected from this mutated hemoglobin protein. This can lead to anemia and other problems. Well, unfortunately, there is not a cure for this disorder yet. The good news is that treatment for this disorder has greatly improved. Another thing to mention, if an individual only inherits 1 copy of the mutated gene from one parent, they are a carrier, but they don't officially have the disease. Usually they do not have symptoms, but those that are carriers appear to have a protective factor against malaria. Malaria is a disease caused by a protist. It can be transmitted by mosquitoes. These individuals can still get malaria, but their symptoms are often less severe. We should mention that studying mutations and genetic disorders is a large and important field right now. Genetic counselors work to help families that may be affected by genetic disorders. If you have an interest in learning more about careers related to this topic, please check out some of our further reading suggestions in the description below. Well, that's it for me, researchers.
Yeah. So something about fruit flies. Yes, they're used in genetics labs all the time. Drosophila, because they're named for your fruit flies. Yes, and because some of their stuff, like with the position and I think size of their wings, the color of their eyes, things like this are very easily controlled. And they're very small, they're inexpensive. But I will say this, they're a nightmare because you have these little tubes and it usually has like this blue medium in them that's a food for them. It's like this pasty stuff. And you just stick all the little larvae in there and they all hatch and then they're all in there. But the problem is you have to open them to get them out, right? Like at some point you got to deal with them and they just go. And you can always tell, like I know when I was doing college biology, you can always tell when some class has hit that point where they're really doing genetics in lab because you're sitting there trying to take notes and those daggone things, they land on your notebook, they're everywhere because they get out. And if you've ever had them in your house, you know what a nightmare they are. So they mentioned bacteria getting mutations and then they don't respond to antibiotics. So this is one of the reasons that it's highly encouraged that doctors not over prescribe antibiotics. Like don't give out antibiotics to people that have a cold because it's a virus and it's not going to do you any good. And if you give it out and you keep exposing the bacteria that we deal with to these antibiotics, you're sooner or later going to find one that has a mutation that makes it resistant. And then that one is going to thrive now because it has a place to grow because it's not getting affected by this antibiotic. So the example of that we hear most about is MRSA. It's mycostatin resistant, staphylococcus, whatever. And that one, that's a big deal. Like if you get MRSA, you can put you in the hospital. That's not great. You don't want it. And then same thing happens to viruses, right? Viruses can get mutations. Everything that's alive is getting, or not alive, anything that has DNA, I'll put it that way, is getting mutations because viruses aren't technically alive. And we call those variations, right? Variations in a species, in a population. So if you think about the variations to a viral population, what is the variant we're all dealing with right now with COVID? It's the Delta, if you're the Delta variant. Okay, so they're naming them alpha, beta, gamma, delta. This is how many we've gotten to. We're past delta now. We're way past delta now. But that is somewhere along the way, there was a mutation. Now that strain is the one that's picked up because it's for some reason, it's more fit than the others to survive in the environment. And if you're building vaccines that work on specific information, if that genetic information isn't in your vaccine, it's not going to work on that variant. So that's why we have to be really careful with trying to keep it under control because it's going to keep mutating. It's going to mutate as long as it exists. Whether or not our vaccines work on it depends on how kind of broad spectrum they are with how much of the proteins they cover and whether you're hitting the right part. Because you just never kind of know what you're going to come in contact with. So genetic control of development. So what controls the development of cells and tissues in multicellular organisms? So this is what we're talking about. Why don't your cornea cells in your eyeballs grow hair? Okay. So you've got master control genes that are like switches that trigger particular patterns of development and differentiation in cells and tissues. If every cell in your body has the same genetic information, then something has to tell your skin cell to make skin, not something else. By the way, it mentioned there are some cells in your body that don't contain the same DNA as everything else. Aside from sex cells, do you know what those would be? What cells do you think your body might produce that don't contain your DNA like that? Well, yeah, that's what we're saying. Aside from those that are half. So it would be your red blood cells, first off. They can get DNA from blood, yeah, but it's not from red blood cells because they don't have the same genetic information. And the other thing would be the cells that your body produces that we think of as dead, like the keratinized stuff, your hair and your nails. So if you find hair at a crime scene, unless that hair has a root or has skin cells on it, it's not going to be useful to you for extracting DNA to catch a criminal or identify a victim or whatever. You have to have something with DNA. Like fingernail scrapings, great. Fingernail, no.
Is all red blood cells have the same DNA?
I believe, yeah, I mean, yeah, they don't have DNA like the rest of your cells do. So it's not like they have the same DNA. They just kind of lack the whole, they're a very different sort of structure. It's kind of, yeah, they're a different class all to themselves. So you've got regulating gene expression is especially important in the way a multicellular organism develops. So think about this. You start as one cell and somehow you become not only billions of cells, but billions of different cells. How do you do that? Something has to code for that. That's also in your DNA. So this right here these are really cool images. I hope you think they're as cool as I think. I love this stuff. So as your embryo develops, you've got different sets of genes that are regulated by transcription factors and repressors. All right, so your gene regulation helps cells undergo differentiation to become specialized in structure and function. You remember a couple weeks back, we watched a video of a cell under a microscope. It got fertilized, it became 2, then it became 4, and it kind of blah, 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 blah. And we got all the way to this morula stage that you could clearly see. And then we get to this early blastocyst where I said it's sort of like a snow globe with a bubble in it, right? It was like, it had like from a cross section, it looks like a basket, but really it's a bubble. This is when this is kind of the last point right around in here when you're not getting major differentiation. When you move on to about 10 days, so this is human embryo, but when you move on to about 10 days post-fertilization, you're starting to differentiate cells. And they're going to differentiate into particular tissue layers that are always organized in the same way. A fertilized zygote is polar, right? Just like you can think of it like Earth. It's definitely, it orients a certain way. And within that orientation, you always have the same setup. So if you took a lateral view, like if you cut it this way and you were to look inside it, all of these different tissues, like right here, this whole chunk of tissue here is all going to turn into external tissue. This whole chunk here is going to turn into nervous system tissue. And then you've got blood, kidney, and this would be dorsal and ventral. So dorsal would mean like your back side, ventral would mean your front side. So this would be like, you could think of it like it's laying on its side almost. This would be a dorsal view. So this is looking at it from the top down. And if you're looking at it from the top down, you'd see all of these different layers. Like this layer is your nervous system layer. This layer is your epidermis layer. Endoderm, this turns into your insides. And this is what's crazy. You'll get to a point where you've kind of got a weird shape, and they have this thing here called a Henson's node that forms. And then there's this sort of a line, and it's really cool to watch. There's some really good, we'll probably look at some at some point. There's some great graphic representations, but there's this gastrulation process where you can actually watch the whole embryo fold in on itself. And when it does that, it's taking all these layers here, folding them in, and it creates two things first. It creates a notochord, which will eventually become a spinal cord, and an alimentary canal, which is eventually everything between mouth and anus, the whole system. And it forms all from these different tissues that have to shift around and differentiate. It's really cool. And like I was saying, like at this point, if you do fate mapping in a lab, you can get a good bioadhesive dye, and you can touch one of these little cells here. And if you're using like an amphibian, we use frogs for it, you can watch that tadpole develop. And this particular, like if you want to hit nervous system, you can find it. You know exactly where it's going to be. And you can hit it and watch that whole frog's nervous system develop in whatever color you tagged. It's pretty daggone cool.
Is it oriented, you said it's oriented polarly, does that mean it?
Yeah.
It has to do with the magnetic polar gravity? of the
I
Earth?
mean,
Or
I
is
think
that-
it does. I don't remember specifically, but I know that, yeah, I think it does. It's obvious when you're
Wait,
looking.
How much would that mess up that level of function?
Right. Yeah. It's more, it's less like thinking about Earth magnetically and more when you're thinking about electricity again. You know, like there's so much electrically going on anyway, because you've got all these, you get down to a molecular level and then you get down to an atomic level. All that stuff,
Breaking and separating.
all that stuff works together, and you can actually watch it orient it very specifically with frogs. You can, if you have a pond or anything nearby, especially in the spring, if you've got standing water, you can actually watch it happen with frogs, because the female frog will lay the eggs and leave them most of the time. Like a lot of species will just lay the eggs and leave them, right? They're just floating on the surface. But the male comes along and it excretes his sperm in the general area. And when those frog eggs fertilize, they rotate. And you can watch, they go from looking like just little jelly balls to little jelly balls with black dots all pointing straight up. They rotate straight up. And you can look at them and go, fertilized, not fertilized. And it's just, it's like, whoop. If you hang around, you can watch it happen. It's not immediate, it's not super fast, but you can watch it happen. It's pretty cool. So then we have homeotic genes. All right, so homeotic genes are any group of genes that control a pattern of body formation during early embryonic development. So this is what we were just talking about. So they have these homeobox genes. It's a DNA sequence around 180 base pairs found within genes that are involved in regulation patterns of anatomical development, so structural development in animals, fungi, plants, numerous single cell eukaryotes. So Hox genes are a subset of homeobox genes, and they're a group of related genes that specify regions of the body of an embryo along a head-tail axis. So that's what's talking about notochord and having, like, you know, which end is which. It's oriented, a head-tail axis. So Hox genes, Hox proteins encode and specify characteristics of position, ensuring that correct body structures form in the correct places on the body. So if you've got a fruit fly embryo, it's got genes that tell it how to become a fruit fly, whereas if you've got a mouse, it tells it how to become a mouse. And these are genes that specifically tell different tissues where to start forming and how to form in relation to one another. So it's super complex, and it's down to this itty bitty granular point. But if you think about it, without that, it would make no sense. You would be all out of order. I mean, you would look crazy. You wouldn't function, right? Nobody would. So we had this dude, Lewis. So from Lewis's work, it became clear that a set of master genes known as homeotic genes regulates the organs and development, specific parts of the body. So if without these, we would be all out of order. Like this is saying, you know, head, thorax, abdomen in this guy, which is pretty easy. And then there's all this internal stuff. For you guys, it would obviously be much more complicated, right? Head, shoulders, knees, and toes. So there are also environmental influences on how your body is organized that your genes will pick up on, not you necessarily, but some bodies. For example, a butterfly. How do you get to the butterfly? You start with the egg, larva, pupa, butterfly. Everybody knows this, right? You learn this in kindergarten. Well, this is controlled by environmental influences, time, temperature, things like that. The temperature and time and all sorts of things are not correct. The genes will not pick up on, oh, I need to start changing now. So you think about this, this is body organization that changes multiple times throughout a lifetime. So these genes are switched on and off based on environmental triggers, without which it wouldn't develop correctly. Another great example are these Himalayan rabbits. Okay, so these bunnies, cute bunny, right? So see the one with the dark ears and the dark feet? All right, now, what part of a rabbit do you suppose gets the coldest?
The ears.
Yeah, it's ears, and what else would get good and cold if you lived in the Himalayas? Yeah, your feet. Yeah, the parts of that rabbit that are, I don't know what, black here in this picture. Why would it be advantageous for that rabbit to have black ears, black feet, black nose? Yeah, what were you gonna say?
I was thinking maybe like the ones before it got like frostbite or something. Like
Yeah,
that.
exactly. So if you want to be, if you go outside in the sun today and you're wearing black, you're going to feel that, right? It's going to be warmer. You're going to absorb all that energy. So these parts of this rabbit's body are affected. Those cells are affected by the environment, and it tells the DNA in those cells produce more pigment. I'm cold. And it produces pigment. And if you rear this rabbit in the cold, he's going to look like this. This is the exact same species of rabbit. And if you don't rear him in the cold, he is not going to turn black. Now, you cool? You look sleepy again. Here's an interesting thing. You can take the same rabbit, say this guy right here. You're rearing him in my house where it's usually about 70 degrees. He's got no reason to be cold. Not kidding. You can look this up. If you strap an ice pack to this rabbit's back and just keep him cold for a long time, keep changing it out, over time, he's going to get a nice big black saddle wherever you strapped it on. Because that part of his body, those
Thermal rabbit.
yeah, because the cells in that part of his body will get an environmental trigger. Ooh, it's cold. We need to produce more pigment and get warm, and it will produce more pigment despite the fact that it's not getting sunlight. It doesn't know.
Amazon has rabbits, right? They have everything.
Right.
Go buy a rabbit. Yeah, go buy a rabbit.
So then you've also got environmental factors and genetic factors that can combine together and create a problem. So say you're genetically predisposed to asthma. Okay. And then say you're hanging out in a place where you've got your allergens or cigarette smoke or smoking. Well, now you've got asthma. So there are definitely different factors that can combine together to cause a different outcome. So some of it's just environment, some of it's environment plus genetics. So it can be, disorders or just differences in general. Can be just genes, environment, or both. And then lastly, this is a great example of both too. So hydrangeas, or this is more environmental really, but hydrangeas, yeah. You ever had somebody change the color of their flowers by sticking like a rusty nail or something in the ground? Yep. Because the soil acidity is what changes the color of those flowers to a degree. Some of them don't change as much. Like they especially now, they've got them bred to be like, this one's pink and this one's blue. And that's because they're taking like kind of your average pH in the soil in an area, and they're breeding them so that they will be that color in that pH, just selective breeding. But yeah, for sure, if you change it enough, you'll get a different outcome. So we got a gene regulation.
I love him.
I know, who doesn't? They're so fun.
I don't think I've ever seen one of their videos.
I could watch them all day. This is a shorter one, but it's a good one. They're addictive. You can go from one to the next. Just these are fun.
These are different amoebas.
Have you ever thought about what a disaster it would be if the cells in your eyes started producing the same hydrochloric acid that is made by your stomach cells? Your stomach cells produce hydrochloric acid to help break down food, but you definitely don't want that in your eyes. Thank goodness that doesn't happen. But it's surprising because both your eye cells and
funny.
your stomach cells contain all of your DNA. Your DNA, all of it, is found in your body cells. But see, the portions that are used need to be regulated somehow. Otherwise, we could end up with something ridiculous like eye cells producing hydrochloric acid. And that wouldn't just be a waste of resources, that would actually be very difficult to explain to your friends. You want genes to be regulated, controlled. Remember that genes are made up of DNA. DNA is used to give instructions for the production of proteins in the process of protein synthesis. But an important concept is that there needs to be a method of determining which genes will be turned on and which genes will be turned off. This is called gene regulation. And there are many ways that genes are regulated. In your human body cells, you can have proteins that can bind to certain gene regions to increase the rate of transcription for the transcription enzyme RNA polymerase. Or you can have proteins decrease transcription to the point that it may not be transcribed at all. This is a form of gene regulation. Your eye cells don't use the portion of DNA that codes for making hydrochloric acid like your stomach cells do, because there is regulation in your cells to determine which portions of the DNA is used. But we want to shift gears now to talk about a very interesting way of regulating genes that can be sometimes a little challenging to visualize. This particular method is really found in prokaryotes, a few eukaryote exceptions. It's called an operon. An operon is a fancy way of regulating genes, and it's usually made up of a few genes that can involve enzymes. Remember that enzymes are proteins with the ability to break down or build up the substances that they act on. Now let's talk about some key players in an operon so that we can see some gene regulation here. First, RNA polymerase. It's a builder enzyme actually, because RNA polymerase is an enzyme. In biology, a lot of enzymes end in that ASE. RNA polymerase is needed in order to start transcription. Remember that transcription
These are
and
so fun.
translation are steps in protein synthesis. Protein synthesis means to make proteins, enzymes in this case. The thing about RNA polymerase, though, it gets a little confusing for RNA polymerase without somewhere to bind. If you watched our DNA replication video, you learned about DNA polymerase and how it had to have a primer to know where to start. Well, RNA polymerase needs a promoter. A promoter is a sequence of DNA where the RNA polymerase can bind. So you would think, okay, you got your RNA polymerase, you attach it to a promoter, and boom, you make your mRNA, which eventually will be used to make a protein. But there's this other sequence of DNA called an operator. The operator is a part of the DNA where something called a repressor can bind. The repressor, if bound to the operator, blocks RNA polymerase.
Oh,
And poor RNA polymerase cannot move forward and no mRNA can be made, therefore no proteins. So let's take a look at our setup here. This is an example called a lac operon. Notice there's a promoter region of the DNA, the operator region of the DNA, and there are 3 genes here that code for enzymes that help in the process of breaking down lactose. Lactose is a sugar. If lactose sugar is around, bacteria want these enzymes to be made so that they can use them to break down the lactose sugar. Then they can metabolize it. Fed bacteria are happy bacteria. Here's the repressor. There's actually a gene here that codes for producing the repressor. See this gene that we call I? It has its own promoter. Well, this gene codes for the production of the repressor. So why do we need this repressor? Well, it's wasteful to make things that you don't need. If there's no lactose, it wouldn't make sense to start making enzymes that work together to break down lactose. It would be a waste. The enzymes would just sit there. So if lactose is not present, then the repressor binds to the operator, and this blocks RNA polymerase. So mRNA cannot be made, and therefore the proteins, the enzymes in this case, cannot be made. But if lactose is around in the environment, something pretty cool happens. The lactose, remember that's the sugar, binds to the repressor. This changes the repressor's conformation. Try as it might, the repressor cannot bind to the operator. RNA polymerase finds its promoter, binds, and transcribes to make the mRNA from the genes on the operon. That mRNA will be used to make enzymes to break down the lactose sugar. And bacteria like to eat, so that makes them pretty happy. say that we think it's pretty impressive to think about all the gene regulation that occurs in cells. And if you find it fascinating, know that there are careers that focus on gene regulation and understanding how genes can be turned on and turned off. Because by doing that, we can gain a better understanding of the variety of diseases that have gene influences in the human body. Well,
So it's it gets pretty involved, but it's really, really interesting. Really, really interesting. So I'll put these up, these last couple here. It's this one, and I think there's like, yeah, one more here, so that you guys can check your answers if you want to see if your notes were good to go. But yeah, it's really, really neat. Next week's lesson is a lot about some of the effects of these genetic mutations and genetic issues, where it's got different diseases, conditions, syndromes, all that sort of thing. Yeah, there you go. Aren't those a real pain? Yeah, so yeah, there's all that. And then yes, well, no, that's what I mean. They can be a real pain if they don't do what they're supposed to do. Otherwise, they're great. So yeah. But excellent. You guys are going to be sure that if you have something to turn in, that you have turned it in. Don't forget to turn in stuff. If you have lab 8, this is it for lab 8, right? So
I think I left mine here. I can't find
Oh, well, now I
it.
have.
yeah, you left them. That it was so funny because you had them. I'd given them to you. And then like that was the day that you and Alex were like particularly goofy and you both forgot your notes. And I thought it was hilarious. I was like, they both left them. Yep.
I'm sorry.
No, you're good. All right.
I could not find my lab 8 anyway.
You couldn't find lab 8 anywhere. It's always online. If you can't find it,
Yeah.
there is a copy
It's just
in
I have
your
my